Hello students, welcome to my educational channel EasyEd. In our last video, we have discussed about characteristics of Kingdom Protista. Now today we will discuss classification of Kingdom Protista. So coming to the classification of Kingdom Protista, you can see here that Protistans have been divided into five main groups. Kingdom Protista is divided into five main groups. What are they? Chrysophytes, dinoflagellates, euglenoids, slime molds, and protozoans. So these are the five groups of Kingdom Protista, and we will study each of them separately. Now first we will take general characteristics of chrysophytes. Now what are chrysophytes? They include diatoms and golden algae. They are commonly known as golden algae and are called as desmids. Okay. Chrysophytes include diatoms and golden algae or desmids. They are freshwater or marine. They can live either in fresh water or they can live in seawater. They are microscopic, as we discussed that protistans are unicellular. It is obvious they will be very small and cannot be seen with naked eyes. So they are microscopic. Next, they float passively in the water currents. Passively in the water currents means, as we have discussed, they are aquatic in nature. So what is the meaning of floating passively in the water current? Means they float along the water current. Along the water current they float, and they are known as planktons. The planktons are a kind of chrysophytes. Okay, examples there. Next, they are mostly photosynthetic. Chrysophytes are mostly photosynthetic. So in this chrysophytes, you have to study about diatoms. So what are diatoms? Diatoms are chrysophytes, and what are their characteristics? In diatoms, cell wall forms two thin overlapping shells which fit together as in a soap box. Means the shape is like that. There are two thin shells and they overlap each other and fit how? Like a soap box. I have drawn a small diagram. You can see here. See, there are shells, okay, and very thin shells are there and they just overlap each other. See, over, and how it is looking? It is looking like a soap case. Okay, they two thin shells overlap each other. Next characteristics, their walls are embedded with silica and thus they are indestructible. Just now you have studied cell wall forms two thin overlapping shells. Means cell wall is forming shell. And how it is forming shell? It should be hard. It must be hard. And how it is hard? The walls are embedded with silica. They have silica deposits. And as it becomes hard due to the presence of silica, these walls are indestructible. Means the diatom is an organism inside the shell. It will die. But because the shell is with the deposition of silica, it remains. It is indestructible. And thus, diatoms have left behind large amount of cell wall deposits in their habitat. Understand? In whatever habitat they live, in whichever habitat they live, what happens? They die on as a completion of their life cycle, but their silicon embedded cell wall, which forms a hard shell like structure, it is indestructible and it remains there. So their habitat has a large amount of cell wall deposits, and this accumulation over billions of years is known as diatomaceous earth. You might have heard about diatomaceous earth. What is this diatomaceous earth? Means in the habitat of diatoms, they leave their cell walls which are silicon embedded, which are shell like structures, and they are indestructible. So there exists an accumulation of these diatomaceous cell walls, as a result of which, after so many years, after billions of years, that particular area is rich in these diatoms cell walls, silicon cell walls. So it is known as, that particular area is known as diatomaceous earth. Being gritty, this soil is used in polishing, filtration of oils and syrups. Means this diatomaceous earth, the place, the soil which is rich in the silicon deposits, hard shells of diatoms. Now what is its use? It becomes gritty, and this diatomaceous earth soil is used in polishing, filtration of oils and syrups. And one important character, diatoms are the chief producers in oceans. In oceans, diatoms are main producers. Just now we have studied that chrysophytes are mostly photosynthetic. Mostly photosynthetic. A plant-like character, as we studied in last lecture, that some of the protistans exhibit plant-like character. So here they are autotrophic, they are mostly photosynthetic, and you might have studied ecology. What are producers? Mainly in every habitat, plants are the producers. But what about diatoms? In oceans, diatoms are the chief producers. Okay? This diagram will let you understand somewhat their body shape. So these are diatoms. Diatoms are grouped under chrysophytes. Chrysophytes are the first group of what? Protistans. Second, we have dinoflagellates. Now, what are dinoflagellates? Dinoflagellates are protistans which are mostly marine. What are the characteristics of dinoflagellates? They are mostly marine, means they are found in salty water or sea water. They are mostly photosynthetic, again autotrophic, mostly photosynthetic. They have pigments present in the cells. Dinoflagellates have pigments. Pigments, you understand by now, responsible for color. So dinoflagellates have pigments found in their cells, and due to these pigments, they appear in different colors. Because of the different types of pigments, colorful pigments present in their cells, these dinoflagellates appear in different colors such as yellow, green, brown, blue or red. In all these colors they are found because of the presence of pigments. Next character, their cell wall has stiff cellulose plates on the outer surface, similar to what? Chrysophytes. Chrysophytes also have silicon embedded cell wall. Okay, here what is there? Cell wall has stiff cellulose plates. Here the cell wall is with plates which is made up of cellulose. In diatoms it was made up of silica. So stiff cellulose plates are present on the outer surface of the cell wall. I will show you a diagram. This is a dinoflagellate, and see, it is a shell-like structure, and this shell-like structure, outermost cell wall, it has what? Stiff cellular plates. These are plates. These carvings, you can see these are plates. So this plates is formed by what? Cellulose deposited cell wall. Next character, most of them have two flagella. Here, as we have studied in the general characteristics of protistans, they have flagella or cilia. Dinoflagellates, as the name is evident, dino means two, flagellates means flagella. Dinoflagellates, most of them have two flagella, And there is a specific arrangement of the flagella. How? One lying longitudinally and other transversely. See the diagram. Two flagella. One lying longitudinally and other transversely. And where? There is a furrow in between the wall plates. In between the wall plates, there is a furrow in which this flagella is there. One is longitudinal and one is transfer inside a furrow. Okay, so these are the general characteristics of dinoflagellates. Diagram I have shown you their specific structure. Now one thing you have to consider here, you have to study here, that very often red dinoflagellates, as we have discussed that dinoflagellates are of different colors depending upon their pigments. So very often red dinoflagellates undergo such a rapid multiplication that they make the whole sea appear red. Red dinoflagellates very often divide or multiply so rapidly. They increase in such a large number that the whole sea appears red. Okay, and this is known as red tides when the sea appears completely red. Why? Because of the large number of red dinoflagellates. There is an example, Goniolex. Goniolex is a red dinoflagellate and it makes the red tides. Red tides means the whole sea appears red due to huge number of Goniolex, that is red dinoflagellates. Now what happens? They are in such a large number that they produce some toxins, and this large number of toxins produced by these large number of red dinoflagellates may even kill other marine animals such as fishes. Huge number of dinoflagellates produce huge number of toxins which can kill the other marine animals. Okay. Then we have our third protestan, that is euglenoids. Euglenoids, what are the general characters? Majority of them are fresh water. Now we have studied dinoflagellates for marine water. Now euglenoids, majority of them are freshwater forms found in stagnant water. You understand, stagnant water deposited at a place, not flowing, stagnant. So majority of them are freshwater forms found in stagnant water. Next, instead of cell wall, they have a protein rich layer called pellicle. Okay, here cell wall is absent, and now this is the character resembling to animals. We discussed in earlier video that some protistans are plant like, such as we studied chrysophytes and dinoflagellates. They are photosynthetic, they are plant like. Now euglenoids, they lack cell wall and this character resembles animal like. That is why protistans are said to be the link between plant animals and fungi. So what is this pellicle? Instead of cell wall, there is a layer surrounding the cell. See a diagram. See, this is euglenoid. This membrane, this covering, it is not cell wall. What is it? It is a protein rich layer. And what is it called? It is called pellicle. And this pellicle makes the body flexible. What is pellicle? Pellicle is the outermost covering of the euglenoids. It is not cell wall, it is protein rich layer. And this pellicle makes the body of the euglenoid flexible. As you can see, they have two flagella. Again, euglenoids like dinoflagellates have two flagella, but difference is that one is long and other is short. Euglenoids have two flagella, one is long and other is short. Okay. Now, mode of nutrition. Euglenoids are photosynthetic in sunlight, but when deprived of sunlight, they behave like heterotrophs by predating on smaller organisms. Now, this is somewhat a specific character. What is the mode of nutrition of euglenoids? If they will get sunlight, they will undergo photosynthesis. But when they do not get deprived, when they do not get sunlight, when they are deprived of sunlight, how do they feed? They behave like heterotrophs. Okay, means they are autotrophic in presence of sunlight, but heterotrophic in absence of sunlight. And how do they feed? They feed on smaller organisms. They predate them. They are predators for the smaller organism. And one thing important you can underline here, note here, that this type of nutrition means both kind of nutrition, autotrophic as well as heterotrophic. This kind of nutrition exhibited is called as mixotrophic nutrition. Means what is a mixotrophic nutrition? Both combination of autotrophic and heterotrophic. So euglenoids show mixotrophic nutrition. You can mark it as important for your exams as well as for NEET. Okay. Now, pigments present in euglenoids are identical to those present in plants. Again, a differentiating character. It does not have cell wall. It has a protein layer covering the body, which is animal-like. But it has pigments. Pigments, and which kind of pigments? Pigments that are identical to plants. That means green color. They show green color. They have pigments identical to the plants. Just now we studied that dinoflagellates have different colored pigments: red, green, yellow, brown, blue. But euglenoids have pigments identical to plants, and they also show green in color, and that is why they are photosynthetic in presence of sunlight, because this pigment is similar to that present in plants. Example of euglenoid is euglena. Euglena, this organism, euglena, it is a euglenoid. Okay. Now next we will study next protestant, that is slime mold. Now again, slime mold general characteristics. Here differentiation starts from the mode of nutrition. They are saprophytes. Okay. You might be knowing what are saprophytes. Saprophytes which feed on dead and decay. So slime molds are saprophytes, and their body moves along decaying twigs and leaves, engulfing organic material. What do they feed on? Decaying twigs, plant twigs and leaves, by engulfing them. So slime molds are saprophytes. And on what they feed? On decaying twigs and leaves. Next character, under suitable conditions, they form an aggregation, accumulation called plasmodium, which may grow and spread over several feet. So, what is this specific character? That when they are having suitable conditions, favorable conditions, what do? They aggregate, they accumulate together. They aggregate and form a structure. See, this kind of structure. These are small slime molds. What is? They have aggregated, they have accumulated and form a structure, and this structure, it is called as plasmodium, and this plasmodium may grow and spread over a several feet. Means many slime molds accumulate together and increase the size of the plasmodium. Then next character, during unfavorable conditions, the plasmodium differentiates and form fruiting bodies. Now here, what are fruiting bodies? Fruiting bodies bear spores. Okay. During unfavorable conditions, plasmodium differentiates. The molds start segregating. And what do they form? They form fruiting bodies, some structures which bear spores. They form spores in unfavorable conditions. These spores possess true walls. The walls of the spores are true walls and they are extremely resistant. In any condition, they can survive. These spores, having true walls, are extremely resistant and survive for several years. They are not destroyed. They survive for several years even under adverse conditions. Okay? These spores are formed during unfavorable conditions. They have true walls and they are extremely resistant, which can even survive under adverse conditions for several years. And these spores are dispersed by air currents, okay, by flow of air, by current of air, by blow of air. These spores are dispersed. Example of slime mold is Physaриum. Okay, example of this slime mold is Physaриum. Now, one thing to note down here and understand well, slime molds. Molds are many times confused with fungi. Okay, so molds are not fungi. See, they are exhibiting fungi like characters. What are they? That is, they are saprophytic. That is, during unfavorable conditions, they form spores. These are all the characteristics of fungi. But these slime molds are not fungi, they are a separate organisms included under protista, and so they are also called as false fungi. So remember this for your competitive exams. For NEET, you can have confusion in that molds are not fungi. Molds, slime molds are protistants and they are known as false fungi. Important thing, remember. Now next protestant. Next protestant is protozoans. Fifth group is protozoans. Now protozoans, they are heterotrophs. Okay, they are heterotrophs. How do they feed? They are either predators or parasites. Protozoans are heterotrophs. They may be predators or parasites. And these protozoans are believed to be the primitive relatives of animals. You know, when we were studying, we studied protozoans under animal kingdom. Okay, but in due course of time, due to vast studies, the continuous changes taking place, what happened? These protozoans, as they were unicellular, they were grouped under protista. So these, therefore, they are, as they are heterotrophs, they do not have cell wall. They are believed now to be primitive relatives of animals because they lack cell wall. Now protozoans, we have four types of protozoans, four groups in protozoans: amoeboid protozoans, flagellated protozoans, ciliated protozoans, and sporozoans. There are four types of protozoans: amoeboid, flagellated, ciliated, and sporozoans. Amoeboid protozoans, each of them you have to study the general character. Amoeboid protozoans, what are they? They live in fresh water, sea water, or moisture. Their habitat, what is that? Maybe fresh water, sea water, or moist soil. Okay. Locomotion is by pseudopodia. You might have heard pseudopodia. How do amoeboid protozoans move? They move by pseudopodia. What is pseudo? Pseudo means false, podia means feet. Pseudopodia means false feet because they are not actual feet, they are protrusions of the cell membrane and used for locomotion, for grabbing food, etc. So this character is there ki protozoans, amoeboid protozoans move with the help of pseudopodia, which is also called as false feet. Their marine forms have silica shells on their surface. Those amoeboid protozoans which are marine, they have what? Silica shells on their surface. Okay. Some of them are parasites. As we have just studied, they are either predators or parasites. Some amoeboid protozoans are parasites, and you know an example, entamoeba. Entamoeba histolytica is a parasite of man. It causes dysentery. Next type of protozoan is flagellated protozoan. Flagellated protozoans are free living or parasitic. Their locomotion is by flagella. As the name is there, their locomotion is by flagella. They have flagella for locomotion. Next, their parasitic forms cause diseases. You know, protozoans are mostly notorious. They cause diseases. They are pathogens. Just we studied Entamoeba histolytica. Now, the parasitic form of flagellated, what is it? Trypanosoma. Trypanosoma is a protozoan, flagellated protozoan, which causes sleeping sickness. It is a disease, sleeping sickness. An example is trypanosoma. Next, we have ciliated protozoan characteristics. Ciliated protozoans are aquatic. Their locomotion is by cilia. As name is there, ciliated. They have cilia for locomotion. Many thousands of cilia are present all over their body, which help in locomotion. Now, one specific character of ciliated protozoans, they have a cavity that opens to the outside of the cell surface. Protozoans, ciliated protozoans have a cavity that opens to the outside of the cell surface, and this cavity is called as gullet. What is it called? Gullet. And what is it for? The coordinated movement of rows of the cilia causes the water laden with food enter the gullet. Now, with the help of gullet, simply they take food. How? With food which is embedded in water, and how? By the movement of cilia, by the coordinated movement of cilia. Okay, example of ciliated protozoan, paramecium. Paramecium is a ciliated protozoan. Now, last protozoan is sporozoans. As the name is evident, they have a spore like stage in their life cycle, and always this spore like stage is infectious because it spreads fast, it causes disease. So sporozoans have an infectious spore like stage in their life cycle, and it is evident that they are disease causing. Okay, example is plasmodium. You might have heard plasmodium vivax. Plasmodium vivax is commonly known as Malarian parasite, and it causes a disastrous disease, that is malaria. Just a fast look on the diagrams of these protozoans. You will see what? Amoeba. Amoeba is an irregular shape, and this is the protrusion of the cell membrane resulting in the formation of a feet-like structure. It is not a true feet, so it is called as false feet or pseudopodia. It helps in locomotion. Then trypanosoma, flagellated protozoan. See its structure, flagella, and with the help of this undulating undulating movement of flagella, it moves. This is flagellated protozoan, that is trypanosoma. Now a fast look on paramecium, ciliated protozoan. Thousands of cilia all over the body surface which help in movement. This cavity which opens to the outside, called as gullet. With the help of the movement of cilia, water taking the food particles enter through gullet, and this is how it takes its food. Example is paramecium. And sporozoans, they are in the form of spores which are infectious. So this was our kingdom protista. Next, we will start with kingdom fungi. Please like, share, and subscribe my channel. Thank you.